So, hello everyone. Good evening. So, we are here gathering on a weekend for for a very new episode of Polaris Talks. I know everybody is so interested to, peek into the new session today. As we have promised you, we have started our academic sessions, and today we'll be discussing about the histology of epithelia. So, before I move on to the whole session as such, myself, I'm Dr. Aishwarya, content lead at Dr. Polaris, and I'll be your moderator today. And for the session, we have Dr. Sanjit Bajpayee, Chief Academic Coordinator at Dr. Polaris, and also Dr. Ryan Fernandez, Medical Director at Dr. Polaris. So, hearty welcome to both of you, sirs. Hope you're doing well.
Yeah, hi.
Hi, Aishwarya. Thank you.
Hi, sirs. So, I'll not waste much of the time because I know much important things are awaiting from you, sir. So over to the presenter today, Dr. Sanjit, sir.
Good evening, everyone. Today, again, we are very fresh with yet another topic in our series of general anatomy. And it happens to be one of the things that happens to bore everyone. Rather, it is one of the topics that doesn't make any sense when you are an undergraduate. That what exactly is the story behind pink and blue? Because when we say histology, we always have a doubt that what is behind the pink, what is blue, and how do pathologists see something beautiful inside a microscope and finally come out to a diagnosis which is as difficult as being spelt as like adenocarcinoma or adenoid cystic carcinoma. This is easily said, right? But it has got a lot of logic behind it. It has got a lot of anatomical orientation behind it. And just in order to tell what you need to know in histology is the basics first. And that is why you need to understand first, then you need to see these slides, and finally you need to memorize or remember them. So whenever we are dealing with a complex topic like histological anatomy, this is the way we have to deal with it. We have to understand, see, and then remember. So let's start today. What I'm going to discuss today, it's about basic histology of the epithelium, right? So let's see what exactly is this. Now, whenever we are a newbie in histological labs, when we are giving slides, we see some kind of this kind of a scenario. Everything is like a dinosaur or an animal. We don't know what is this teeth, what is this eye. It seems like a very, a nice figure under the microscope, but it makes no sense. But then finally your teacher comes inside the class and then they say, can you observe the nuclei? They are flattened. Can you observe the cytoplasm? And you are clueless. Where is the cytoplasm? Where is the nuclei? And what this figure actually makes sense or not, right? So this is for beginners. But by the end of this particular webinar, I hope that you will start seeing the story behind this dinosaur and what exactly is there making it, right? So let's begin. So the initial part that comes in your mind whenever you're doing histology is that whenever you put your eyes under the microscope, you see something like this, right? Something is blue, something is red, and it makes no sense at that particular point of time. Rather, that image gives you vertigo. I feel giddy after seeing so much of red and blue and white, and that finally I don't know what it is, and finally my teacher comes and says, no, this is glandular epithelium. Now finally I don't know why did that teacher say that. So in order to make things simple, whatever you are viewing under the microscope, they are all cells to begin with. Now, if you take out one cell from here, you can see this is the cell that we have here, right? Now, the cell, as we know, it is made up of a cell membrane outside, and it has got something called as a protoplasm inside, which is again divided into cytoplasm outside, and in center, there is a nucleus. So this is basic that we already know from our cellular biology knowledge, right? Now, when we are talking about this, we know that there are two segments to it. One is the cytoplasm. This cytoplasm contains all the organelles as well as it contains various kinds of proteins. And then it comes inside the nucleus, which contains all the genetic material. Now, why this is important? Whenever you are putting a stain to a cell, it will stain differentially. It won't stain together. Some stain will be taken up by some part of the cell. Some stain will be taken up by some part of the cell. So some parts of the cell will appear to be darker and some parts of the cell will appear to be lighter, what we say as pink and blue. So this is what exactly is happening. Now, when we realize what is a tissue? Tissue is a group of cells. So whenever we are taking a section of a tissue and seeing under a microscope, we are seeing these millions of cells getting stained by two important dyes, right? So the most commonly used dyes or the most commonly used methods of staining these cells are something present here. And these are called as H and E staining, or what we call as hematoxylin and eosin stain. Now, this has to be very clearly understood. These are not the just two dyes that are present in pathology. There are various dyes. There are, various kinds of structures which stain with different dyes. But the crux or the main dyes that are used in everyday staining of tissues are these two. So you have to master H&E first, and then later on, when there are certain tissues, you're talking about lipids, there are certain other dyes, or when you're talking about certain tumors, there are certain other dyes. But these are the stains you need to master upon. Now, why this is red and why, or pink, or why this is blue? Now, this is the concept, okay? So when we see first eosin, you see the bottle is pink. This dye, okay? So you see 1% eosin, this is an acidic dye. Now, when we say acidic, it is negatively charged. What is present in the cell? I told you there are two parts, cytoplasm and nucleus. Cytoplasm has got a lot of organelles. It has got a lot of proteins. Those are positively charged. So eosin, which is acidic, negatively charged, will bind to the cytoplasmic proteins and organelles, which are positively charged, giving the cytoplasm of the cell pink color. So remember that it is not the nucleus which stains pink. It is the cytoplasm of the cell which stains pink. Now, what happens with hematoxylin? If you see, this bottle is blue, right? So this is a basic dye. So blue and basic, B and B. So basic dyes will attract what? They will attract something which is acidic, right? So inside the nucleus, there are certain negatively charged molecules. And outside, they are positively charged. So if you place a basic dye in a nucleus, what will happen? It will take the dye. So in specific, what I told you was, eosin is acidic, it will stain cytoplasm. Basic is hematoxylin, it will stain nucleus, right? And there are also some things called as ribosomes, which also take up this part of blue. So they also stain blue, right? So this is the basic why the pink and why the blue happens in general histology. Okay, now let's move on to the topic per se. So if you see, what exactly is epithelium? So epithelium, this is the word, right? So initially, when epithelium was seen, it was broken down into Greek language into three particular terminologies. Epi means something above. Theli was initially called as a nipple, but it is taken as a structure. And is the aggregation. So if you see something above a structure which is aggregated is called as epithelium, right? So again, this is a singular word. If we call a plural word, this is called as epithelia, right? So epithelium is again singular. So as I told you, it is one or more layer of cells that are covering the outer surface of the body or the outer surface of a particular structure or the inside of a particular structure. So inside or outside, you have epithelium everywhere. Now let's say this is a person who has a beautiful skin outside, but if we actually look at the picture in a microscope, it appears to be having this. It has got multiple layers. And what are these multiple layers? These are aggregations of cell which are forming a very important epithelium, which is called as the epidermis, right? So this is the epithelium that is lining over on the skin. Now, just beneath the epithelium, there is something called as a connective tissue. And I'll come to you later on, but remember that there is epithelium above and there is connective tissue below. And connective tissue will be dealt separately in histology. So we are not going into the connective tissue part. We are only dealing with the lining today. We're dealing with how these linings are formed, right? Now let's see what exactly this lining is. So I told you, when we are talking about epithelium, this epithelial lining is present either on the surface of a structure or inside the surface of the structure. Now let's suppose this is an esophagus or a food pipe. This particular esophagus is also lined from the outside by a particular epithelium. It is also lined inside by a particular epithelium. So now let's say when we are dealing with the epithelium on the outside, it is called as surface lining or surface epithelium. And one type of epithelium is lining the lumen of this esophagus. That is called as luminal lining or luminal epithelium. So this is the difference, and this is where the concept comes, okay? Because this luminal lining is very, very important to understand, because the epithelium might be different here and the surface might have a different epithelium, right? So let me take you more into it so that you can understand, right? So now here you see a kind of a structure, and you see that I've taken out the esophagus and I've given it to you here. And when you see this part of the food pipe, let's suppose this is the food pipe, this is the surface lining or the surface epithelium, but the inside is the lumen, and the cells which are facing the lumen are called as luminal epithelium. Okay, now let's take out this part of the lumen and see in a bigger picture. So this is the bigger picture now. What we are seeing, that these cells are arranged in a group. Okay, this is the first thing that we see. Second, what we see is that one part of the cell is towards the lumen, which is here. This is called as the apical part of the cell, or the apical membrane to be specific. So this part which is facing the lumen of the cell, this is called as the apical membrane of the cell. And one part of the cell is resting behind. That means it is towards the surface. This part of the cell is called as the basement membrane, or the basal part of the cell. Okay, so one part of the cell is called as an apical part, one part of the cell is called as the basal part, which rests upon a membrane called as basement membrane. Very, very important, right? Okay, now once we know about the apical membrane and the basement membrane, let me take about the features of epithelial tissue. Now, what are the features of epithelial tissue? Number one, they are very cellular. So I told you, tissue is a aggregation of many, many cells. So this you can see they are stacked up together, so they are very cellular. They are less vascular. Okay, so always remember, the blood supply of these particular cells lie beneath the basement membrane, in the connective tissue. I told you that there is something called as a connective tissue below this basement membrane, which contains all the capillaries and all the blood vessels, as well as the framework of the cell. But above, there is very less vascularity. So this is the second feature of epithelium. They are very cellular and they are less vascular. Third, I already told you that they are resting on a membrane, and this membrane on which these cells rest is given the name as basement membrane. Okay. Now, these cells show polarity. Now, what do you mean by these terms? Okay, let me clear these terms here itself. Polarity means how the cells are particularly oriented to each other. So there are certain, proteins, there are certain organelles inside the cell. These are distributed all throughout the cell, these organelles, these proteins. Now, wherever these proteins and organelles are going, they are basically changing the shape of the cells accordingly. So if you see, they have a particular orientation. All of these organelles, all of these proteins are oriented equally in all these cells. And that is why they are having the same polarity. Now, let's suppose if a person has a tumor or a cancer grows fast, right? One above the other. These cells will become more and they get stacked up. Some cell will be small, some cell will be big, some cell will be absolutely out of shape. So they won't be symmetrical. So what we will say, they don't show polarity. Okay, that is the meaning of polarity, that how symmetrical they are. And then is they have certain surface modifications. Now, what do you mean by surface? I'm talking about this surface. So on these surface, they can have certain kind of modifications or certain kind of things which are present here. I'm going to deal with these modifications separately. Okay, so these are the general features of epithelial tissues. Fine. Now we move on to the classification of epithelia. This is how we are going to deal with it. Okay, so you can divide epithelial tissue classification in different ways. If as per the function, as per the shape of the cells, as per the number of the cells, you can divide it separately. But the first type of classification is on the basis of their function. Okay, so first we see what we are reading today is the surface epithelium or the lining epithelium, and they are mostly for two important functions: protection and absorption. These are the main crux of these things, right? So this is the first part. Second type of epithelium is something that is glandular, gland-like. You can remember the glands. What do the glands do? They secrete. So second thing is secretion by glandular epithelia. Third type is something which is related to neuro, right? Neuro means sensation. So some epithelia which is related to sensation, we call them neuroepithelia. And then comes something which is contracting and relaxing. That is what? The muscle. So muscle is myo. So myo just attach epithelia to it, so it becomes myoepithelia, right? So these are the classification of epithelia that are solely on the basis of, again, function, or what exactly they are functioning as. Now, I'll tell you the concept behind this particular cell and the basement membrane. Why this basement membrane is important? Then once you know all these things, you can actually, join these things together and finally start reading the slides easily. But you need to know what exactly is there. So you know now why there is pink and blue. You know what is an epithelia. You know what is an apical and a basal membrane. And now I'll tell you exactly what the basement membrane is all about, the concept behind it. Okay. So I've already told you that each of these cells are symmetrically stacked together. Okay. And they have got two important surfaces. One is that is towards the free area or towards the lumen, and the other one is towards the base. The area which is above, which is projecting outwards towards the structure, is called as an apical surface or the apical area. And the other one, which is towards the base, is called as the basement membrane, right? And I told you again that below the basement membrane is the connective tissue. So you already see this is the connective tissue, and you can already see there are multiple blood vessels inside it. So that is why I told you epithelia is not itself vascular. It is a connective tissue which contains all the blood vessels. Okay, fine. Now this basement membrane, what does it do? Basement membrane is a cement. Cement as in it is connecting two important structures, one structure above, one structure below. So if you see, the above structure is the cell itself and below structure is the connective tissue. So what exactly is the sealant in between them? This is the basement membrane where the cells are resting. Okay. Now that is first point. Second point is this basement membrane acts as a physical barrier. If you see, this is not going to let anything from the outer surface of the cell freely percolate into the connective tissue. It is acting as a mechanical barrier. So none of the high level proteins or none of the things which we don't want can enter inside through the cells into the connective tissue until and unless we don't want them. If we want them, we have given a specific channel for that. Okay, this is the beautiful part about basement membrane. It's a protective barrier. Third, this basement membrane is again divided into two parts. One part is the one which is above, which is towards the basal part of the cells. So it is named in the same way, basal lamina. Lamina means a sheet in literal terms. So one part is towards the basal part of the cells. This is called as basal lamina. One part is towards the connective tissue. This is called as reticular lamina. And this is what the basement membrane constitutes. There are two parts, basal lamina and reticular lamina. Now, just to make things familiar to you, if suppose this person has got a cancerous growth, what will happen? Cells will keep on stacking above and above and above, because cancer is something that is abnormal proliferation of cells. So finally, what will happen? Once the cells will grow more and more, after one point of time, this basement membrane will not be enough to actually take them. And this basement membrane will give away. So what will happen? All the cancerous cells will come down and they will go into various structures which we don't want. It will metastasize. So our basement membrane is very important to protect us. And that's why it is protecting all our cells in this particular fashion. And it is sealing two particular important areas. Right. Now we go on to what are the functions of these kind of epithelial tissues. Okay. So I already told you the important functions are divided as per the epithelia. There are different epithelia in different parts of the body. So protection, absorption, secretion, exchange. These are the four important features of any epithelial tissue, how they are present or wherever they are present. We are going to see that, right? Now, let's say this is particularly a man who's scratching his neck and he has got some kind of a skin allergy. So what has happened? An allergen has just poured over here something and he has started to scratch it. Now, why didn't that allergen freely go inside and go everywhere? Now you understand. It's the protection mechanism by the epithelium that is giving it. So it stays over on the surface and you can easily scratch it off. Or you can easily remove it off, right? Similarly, there are certain epithelia which absorb and release something. There are certain epithelia which secrete something. And there are certain epithelia which help in exchanging of different materials across, right? These are the major four functions of any kind of epithelium. Right. Now I move on to a different classification of epithelia. I told you one of the epithelium classification is based on the functions. There were four functions. I told you how these epithelia are divided on the basis of function. Now comes another classification of epithelia according to the number of layers. Okay, this is very, very important. If there is a single lining of an epithelial tissue, we call it simple epithelium. Just remember that. So if there is only a single lining of a tissue, we call it simple. Let's keep it simple. If this Simple epithelium gets stacked with more cells above. That means now it is not unilayered, it is multi-layered. Now anything multi-layered is given the name as stratified. So we call it stratified epithelium because it has got a number of layers which are more than usual. And third comes pseudo-stratified. Pseudo means false. Stratified means multiple layers. That means they are a single layer of cells, but they look that there are multiple. Now, I'll tell you later on why this happens. But just remember that pseudo means false, stratified means multiple layers. That means that a person is seeing multiple layers, but actually there is only a single layer of cell. That is called as pseudo-stratified. Okay, so on the basis of the number of layers, we have these simple, stratified, and pseudo-stratified layers. Okay, now comes on the basis of their shape. This is the third classification of epithelium. So on the basis of the shape of this, they are again divided into majorly three types. That is squamous, cuboidal, and columnar. Now, what do you mean by this? Squamous, literally in the word, that is Greek word, means flat. So if you see that all of these cells are like flat or flattened. They lie on the surface and they are flat. So what do we call them? We call it squamous. Okay. Now, if you see the other kind of cells, these are cube-like. You can see these are cube-like. So we call it cuboidal. Similarly, if you see these cells, these are in the form of columns. So what do we call them? Columnar. Okay. So squamous, cuboidal, and columnar. These are the three types on the basis of shape. Now simply add one thing to another. If I have one layer of flat cells, what do I call it? I call it simple squamous epithelium. If I have multiple layers of flat cells, what do I call it? Stratified squamous epithelium, because stratified means multiple layers. Similarly, cuboidal becomes, if I have a single layer of cuboidal cells, what do I call it? Simple cuboidal epithelium, because simple means one layer. Similarly, if we have stacked above many cuboidal cells, what does it become? It becomes stratified cuboidal epithelium, correct? Now comes the column part. If you have a single layer of column-like cells, it is called as simple columnar epithelium. And at the same time, if they are stacked one above the other in the form of columns, we call it stratified columnar epithelium. Pseudo-stratified columnar, I'll come later when I deal with them separately. Okay, so just remember, on the shape, on the size, and on the basis of the function, you have three categories in which you can differentiate these kind of epithelia. Okay, now let's take up one epithelium at a time, and I'm going to show you various examples and how to see these slides and how to actually, take knowledge of what has happened to the patient. Okay, so the first epithelium is simple epithelium. So I already told you what is simple. Simple means single layer. Okay, that is having something or a layer of cells which is in a single row. So we call it simple epithelium. Now, simple epithelium, the first category of epithelium under simple epithelium is simple squamous epithelium. Now, you already know what is squamous. I already told you. Squamous means literally flat. So if you see these kind of cells, they are all flat. Now, this is the surface view of these cells. Now, what does it show? If you see the squamous epithelium from the surface, you will see this pavement-like structure. It is like a pavement. You must have seen on a footpath when you walk. It seems like a pavement. Okay, you are seeing multiple polygonal cells which are pavement-like. This is the word. Now, the second thing is, if you see this particular cell from the side, you will see that these are having these kind of flat appearance. And because they are flat, their nuclei is also flattened and it is present at the surface. Okay. Now, because the nuclei are flattened and present at the surface, they usually give this particular epithelium a dome-shaped look. That's why if you see these epithelium are one dome, another dome, another dome. So if you see from the sides, these flattened nuclei are giving them a dome-shaped appearance, or they are giving them a kind of a hoppy appearance, just like you can see a speed breaker coming in. So this is the kind of appearance you get in squamous or simple squamous epithelium. Similarly, what is the basement layer called? This is called as basement membrane, and below that is the connective tissue that I already told you. So now you understood. This is about how these look from the top, and their nuclei is towards the periphery and it is flat. This is how squamous epithelium will look like. Okay, fine. So if you see this particular area, okay, let's say this is the area where you have taken a histological slide and you're seeing these particular epithelium. How will you read that? Now you already know, see, this is the flattened nuclei that you're seeing at the edges. right? And it is flat. And you can see these are like hip hops. They're giving a kind of a speed breaker like pattern, right? So what do you call that? This is a lining of simple squamous epithelia. Simple, right? This is a single layer, so it becomes simple, right? Now, where is this particular location, or where is this particular simple epithelium or simple squamous epithelium found? So this is a very important MCQ based question, because you will get multiple choice answers and you have to choose one of them, right? So in order to remember, think mnemonic wise. Okay, you understood, I told you, you understood. Now you have seen, now you memorize what exactly and where is the location of this particular epithelium. Now just think, when you have got structures or cells which are stacked up together, they are not letting anything go from above, and they are protecting things, right? Okay, so they are basically the protectors. That's why everything in the body which is lined by something which is very, very important is lined by simple squamous epithelium. The lining of the body, the lining of different organs of the body. Now let's say this is a mnemonic called as blame, okay, where you can understand what exactly is the lining of squamous epithelium. B means Bowman's capsule. Now again, Bowman's capsule is a part of a nephron. It is a part of a kidney, which helps to take out all the unwanted material that you don't want into the nephron to come. So it is acting as a protective barrier. It is not letting unwanted proteins to come through it. So what is it happening? It is basically filtering things out, right? That's why B is for Bowman's capsule. L means lining of important structures. I already told you, simple squamous epithelium are the liners of something very, very important. Okay, so you already saw B for Bowman's capsule, that is inside the kidney. The second lining will be of important structures. If they are lining the lung, they are called as pleura. If they are lining the heart, they are called as pericardium. If they are lining the abdomen, they are called as peritoneum. They are all lining very, very important organs inside the body, right? That is why they are the protective barriers. Now comes the next part, alveolus. A. Alveolus is basically nothing. It is a small unit that is present inside in the lungs. There are many, many alveoli which are present inside the lungs which help us breathe. Okay, I'll show you what exactly they do. Now, M is the mnemonic mesothelium. So mesothelium, don't forget, all of these lining of important structures like pleura, pericardium, and peritoneum is called as mesothelium. That's why I've kept M separately. So a very famous mnemonic that comes is, which of the following structures is lined by mesothelium? We usually ask you. So you should not confuse pleura, pericardium, peritoneum. They are all collectively called as mesothelium because they are lining important structures. Okay. And then comes E. E means endothelium. Again, endo means inside. Inside something, what? Inside the capillaries. So they are inside the capillary lining also is simple squamous. And inside the heart, there is something called as endocardium. That is the inside lining of the heart. That is also formed from simple squamous epithelium. So don't forget this mnemonic name. B is for Bowman's capsule, L for lining of important structures, A for alveolus, M for collectively called as mesothelium, and E is endothelium and endocardium. Okay, so this is how you can recapitulate faster when you are actually giving exams. Now let me show you. Right, so when we move on to this particular thing, I told you that this is an alveolus. Let's say this is an alveolus. I've taken out an alveolus from the lungs. Okay, and you know that there are certain arteries in the lungs which carry oxygen. Okay, there are certain capillaries which are carrying oxygen, and this oxygen is going inside the lung, right? The oxygen is coming outside from the lung inside these capillaries, and carbon dioxide is released back. This is what is happening. So on one side, the oxygen is basically traveling from the alveolus into the capillaries. On the other side, carbon dioxide is coming back and you are exhaling out carbon dioxide. This is a regular mechanism, right? So now in this particular diagram, you can see the function of squamous cells. They act as filtration barriers, right? They are causing selective exchange of gases between two barriers. Now simply understand, our capillaries are lined by endothelium. That is also a simple squamous epithelium. Our alveolus is lined by simple squamous epithelium. They both cause exchange of important gaseous material, and finally you are able to breathe. Okay, so this is what exactly the simple squamous epithelium is all about. Remember the mnemonic blame, right? Now let's come to the second part of simple epithelium. I told you simple squamous means simple flat epithelium. Simple flat epithelium is again, the second will be simple cuboidal epithelium, cube-like. So simple means on the basis of the number of layer, that is single. Cuboidal means that it is the shape of the cell that is present there. So let's see this particular cell. This particular cell is cube-like. Now, if there are multiple cube-like cells together, they will be called as simple cuboidal epithelium. Just see this particular image. Now, what do you see in this image? In this particular image, you see that the nucleus is in the center. So the nucleus of these particular cells is in the center and it is round, right? This is the first characteristic. Second, the height as well as the width of these cuboidal cells is the same. That's why they give a cube-like appearance. That's why when you see them from the top, you see multiple cubes there, right? Now just see this image inside a microscope and you will see that this microscopic image, you can see these kind of cells like this. See, their nucleus is in the center, which is staining blue, and around the height and the width is the same. And the cytoplasm is pink. This is a cuboidal epithelium. And you can see it is layered, so it becomes simple cuboidal epithelium. Correct? Fine. Now we move on to what is the location of this cuboidal epithelium. So in order to mention or in order to remember, here also you have a mnemonic. Just remember the mnemonic red clot. So a clot is always red in color. So remember this mnemonic red clot. Now, how do you remember this? Okay. Red means R. R means retina. Retina, the pigment layer, that is also something which is colorful, pigment-like. Okay. So retina pigment layer is lined by this kind of epithelium. Okay. That is R. Then comes CLOT. C means choroid plexus. Now, always remember one thing. These kind of epithelia are mostly secretors and absorbers. Remember this line. They are secretors and absorbers, cuboidal epithelium, right? So when we have choroid plexus is a plexus which is present in our brain. It is present in the ventricles of the brain. There's something called as ventricles. These ventricles are the ones which are containing our cerebrospinal fluid. So choroid plexus is the one which makes this cerebrospinal fluid. They are secretors. So I told you, cuboidal again will have this kind of secretions. So secretions of what? Of, again, cerebrospinal fluid. So choroid plexus. Then comes again L. L is lining inside the lens. Lens is in the eye, right? So that becomes lens. Another thing is O. O means ovary. So the surface of the ovary is also covered with this particular epithelium, a simple cuboidal epithelium, because it increases the surface area there. Okay. And then comes T, that is thyroid follicle. So I again told you secretors and absorbers. So thyroid follicle secretes thyroid hormone. Right? So that is why they also are lined by simple cuboidal epithelium. Right? So now you understood. Now you memorize it. So how do you memorize it? Remember the mnemonic red clot. Okay? Retinal pigment layer. Again, choroid plexus. L for lens. O for ovarian surface. T for thyroid follicles. This is the mnemonic behind simple cuboidal epithelium. Okay? Fine. Now we move on to the third simple epithelium. Okay, so don't get confused. I'm still in the simple part. Simple means one layer. And according to the shape, I have covered what is squamous. I've covered what is cuboidal. And now I'm covering simple columnar part. So I already told you what is columnar cells. Columnar cells are like big columns. So if you see the characteristic of these cells, these particular cells have a nuclei which is elongated and it is present towards the base of the cell. Now I told you cuboidal has a round nuclei in the center. But these particular nuclei are at the basement part or towards the basement membrane, you can say, to be specific. And they are elongated. But always remember, the basement membrane does not touch these nuclei. They are away from the basement membrane. They don't touch it, but they are towards it. Okay. Simply, many columnar cells stacked together will form a simple columnar epithelium. Right? Now you see, the height of these cells is much more than the width of these cells. This is the second point. Why columnar cells or how to identify columnar cells. Okay. So if you see particularly this image, okay. So once you're starting to read slides or histology, the first thing you should see is what exactly is the shape of these cells. Okay. So you see, okay, there are multiple cells which are elongated and they have their nuclei which is elongated towards the base. The first thing comes in your mind, okay, this is columnar epithelium. Now, why do I call it simple? Because it is a single row. So it becomes simple columnar epithelium. As simple as that. Okay, fine. Now, this columnar epithelium is the one which confuses people a lot because there are a lot of terminologies, there are a lot of things inside this columnar epithelium, but it is very simple if you understand it once. Okay, now let's dig into what is columnar epithelium. So let me take you back to the slide again. Okay, now I told you in the beginning that these particular epithelia have got 5 features, I told you. in which one of the features after polarity was surface modifications. So what do you mean by surface modifications? That means on top of these cells, towards the surface or towards the apical membrane, these particular cells can have a kind of a modification, or they can have a kind of a feature which will modify them from the simple part, that they are now something which is different. Okay, these are called as surface modifications. Now, how can a cell become different? These surface modifications are of three types. Okay, one, this particular thing above the apical membrane can project out like multiple finger-like projections. These finger-like projections are given the name as cilia. Now, what do you mean by cilia? Cilia are nothing. You know what is an escalator, right? You've seen an escalator. What does an escalator do? You stand on one side and you reach the other side by just standing there. Cilia are our escalators of the cells. What do they do? If cilia are present like this, just look at my hand. What will happen? Once any foreign material comes here or any mucus comes here, they will move in a coordinated fashion and come back. So they are basically movement. They are propelling and then they are coming back. Again, they are propelling, they are coming back. So this is like a conveyor belt, what we say. Like once we keep our luggage on that conveyor belt, it will lead to the other side. You understood? So cilia are providing motility to the cells. This is the major part. So some of the cells, some of the columnar cells have motile cilia over their surfaces, which makes them motile, which makes these cells motile. Okay. Now, there is another thing. This is called as microvilli. Now, micro means small, villus means a projection. Just like cilia, microvilli are also projections, but these projections are non-motile. This is a major difference. So if you see the cilia are much longer. The length of the cilia is approximately 10 micrometer. But this particular thing is very, very small. So it is very, very minute. It is 1 to 2 millimeters. So it is extremely small. So smaller forms of cilia are called as microvilli. Now these microvilli, what do? They are non-motile, but if they are present, they will increase the surface area of the cell. They will increase the absorptive capacity of the cell. That means if they are present like this, they will increase the absorption that is happening in the cell, as well as they will increase the surface area of the cell. And I'll tell you exactly examples of where you will find these, right? Okay. Now, if you just mix two microvilli together or stack two microvilli together, they are given the name as stereocilia. Stereocilia also are non-motile. They happen to have the same function. Okay. So stereocilia is microvilli put together. Cilia is the motile part. Microvilli and stereocilia are non-motile. This is the difference between the two. So now you understood. This is what surface modifications of the cell is all about. Okay, fine. Now I'll tell you some clinical examples as to what it is. Now I told you about microvilli. These are like projections which are coming out. Just remember this brush. Now when you are brushing, okay, this is a very, very important MCQ question. That what is striated microvilli, what is brush border microvilli. So just remember your toothbrush. If you have these microvilli which are coming out together from the brush in a straight fashion like this, multiple. So see this, these are all straight, stacked together. This is called as a striated border of microvilli. But if it is in a zigzag fashion like this, where toothbrushes come in a zigzag, this is called as brush border. So this is a very important MCQ. What exactly is brush border? Brush border means that the microvilli are against each other. They are coming like this. But if they are straight and coming and pointing, they are called as a striated border of microvilli. Okay. Now you must be thinking there's so many things that I've already told you. It will make sense now, right? Fine. Now you see this. You see this? You see this particular layer of cell. This is a columnar cell stacked up together, which is a simple columnar epithelium. Okay. They have got elongated nuclei. Now you see the other end of this. The other end has these kind of projections which are coming out, which are multiple. And they are much larger than the normal. They are causing motility of the cell because they are moving like this and this. So if any particular dust, any particular mucus, any particular foreign body will come, this will keep moving like this, just like my cursor, and it will finally flow out. This is what is called as cilia. Now I'll give you a clinical scenario to that. In particular people, there is a syndrome called as immotile cilia syndrome that you'll see later on. What happens in that? The cilia don't work. Now, if these cilia don't work, what will happen? The mucus inside the lungs will keep collecting, right? And what will happen? There will be so much of cough, there will be so much of expectoration, and the person might end up into pneumonia. because there is so much of secretions inside, because the cilia are not working. Similarly, if you see the cilia in a uterus, okay, in a uterine tube, what we call as a fallopian tube, if these cilia are not present, they will not let the ova come from the ovary inside into the uterine cavity. They won't. And that is the reason what exactly these cilia are doing. They are just pushing things from one side to the other. And if this particular epithelium is lined by cilia, we call it ciliated columnar epithelium. As simple as that. Okay. Now you see the other picture. This is also a lined cavity, right? Lined epithelium. This is also simple columnar. But you see that again, the border is much more tiny than these cilia. This border is very, very tiny, and this is just like what you have like a brush. And that is why this is called as a brush border epithelium, or if it is straight, then it is called as a striated epithelium lined by microvilli. You understood, right? So this is what exactly surface modifications are all about. Okay, fine. Now we move on to something that is present in between columnar cells, which is very, very important. So you must have seen this particular picture. Let me take back. And you see that these are the normal columnar cells, but you see something vague in between them. See, this is something present between columnar cells. I'm not talking about squamous. I'm not talking about cuboidal. In between columnar cells, there are certain specialized cells or a specific type of columnar cells. These specialized cells are just like a goblet. You know the goblet of fire, like Harry Potter's. There was a goblet of fire cup. It was like a cup which was projected upwards. This is just like a cup. Okay, and this is called as a goblet cell. Now, what does a goblet cell do? A goblet cell is basically having some kind of secretory vesicles. Okay, so this is a cell. It is just like a normal cell, but what does it have? At the apical part of the cell, there are certain secretory vesicles, or it is filled with certain mucin secreting balls which are present there. Now, once this ball of mucin will go ahead from here, it will release mucin. Correct? Now, just imagine, where do you require mucus? You require mucus for lubrication. Now, where all do you require mucus? You require mucus in your respiratory tract, so that all of your dust particles don't stick here and there. It is very much important to lubricate your lungs. Correct? You require mucus inside your intestines, right? In order to make your food and food particles more viscous, so that everything goes out. Otherwise, everybody will end up into constipation. Correct? So you require these kind of goblet cells everywhere, wherever there is some kind of a logic that we require mucus or something wet to be secreted. That is the logic behind it. So you see, this is like a goblet. That's why it's called as a goblet cell. And it contains secretory vesicles which contain mucin. And these particularly come out from this part of the cell. So if you see the previous slide, these are the areas where goblet cells are. And if you see, they are all opening up in the surface epithelium. And they secrete something called as mucin, and this mucin lubricates the whole lining. Are you understanding what is the importance of goblet cell? This is the importance of goblet cell. Right. Now, where do you find this columnar epithelium? Let me be now coming to the memorizing part. You have already seen that how these are and what exactly columnar epithelium is all about. Now, you know what is simple columnar epithelium? I told you simple columnar epithelium is a columnar epithelium which is a single layer lined by columnar cells in a single row. Now, if you have simple columnar without any surface modifications, that means without any brush border, without any cilia, this is usually seen in the stomach and the large intestines. And if you see, goblet cells are a lot. That is why in large intestine, you require a lot of these goblet cells to secrete that mucus. Sometimes you see that a person has passing stool and they see a kind of a mucus kind of coming out. Where is it coming from? It is these goblet cells. Right? Now you understand the logic behind it. Right? So simple columnar epithelium without any surface modifications is usually seen lining the stomach and large intestines. Okay. Now comes, where do you find this cilia? I told you surface modification is ciliated columnar epithelium. So where do you find that? Remember tubes. Now wherever tubes come, tube means these are particular cavities which need cilia to move things here and there. So first cavity is the respiratory tract itself, which requires cilia so that everything foreign is expelled out. Okay. So that otherwise everything or a dust particle will just go inside and sit inside your lungs. It is expelled out by these cilia. Second is the uterine tube, or let's say a fallopian tube, right? So that is also lined with this particular epithelium, which is helping the ova come towards the uterine cavity, right? The uterus itself is lined by this epithelium. Then there is something called as an efferent ductus of the testes. So basically the testes are producing sperms or spermatozoa. They have to travel, and these efferent ductus are going into something called as a vas deferens. Vas deferens is nothing. It's a channel which is basically connecting the testicles to the main ejaculatory duct. So this is just letting everything travel out, the spermatozoa to travel out. Then comes eustachian tube. Eustachian tube is nothing. It is a tube between the ear and the nose. And you don't want anything foreign to come from the nose into the ear. So it requires cilia. Correct? At the same time, ventricles of the brain, what I already told you, that ventricles are something which are present inside the brain and they have CSF or cerebrospinal fluid. Cerebrospinal fluid has to move. And how will it move? Through cilia. Correct? So ventricles of the brain are also lined by this epithelium, and there is something called as ependyma of central canal of spinal cord. Big word, but just remember, in the spinal cord there is something called as a central canal. There in the center of the spinal cord there is a small canal, and this canal is meant for CSF circulation. So the lining of that is called as ependyma, which is a glial cell, and this ependyma is basically helping that CSF circulate up and down. It is maintaining CSF circulation or maintaining homeostasis there. Right? So now you remember the tubes. If you remember the tubes, you'll remember ciliated columnar epithelium well. Right. Now, again, the last part of columnar epithelium. Some part of columnar epithelium, I already told you that they have a brush border or they have a striated border. Now, what do you mean by striated border? That they're lined by various microvilli. Now, where do you find these microvillus lined columnar epithelium? You will find it in small intestine. Okay. Now, I told you that wherever there is a striated border, what does it do? Or a brush border or a microvillus, what does it do? Microvillus has increasing of the surface area. It is a good absorber. If surface area is increased, something will absorb more, right? So that is why small intestine in churning of food, in absorbing the important things, in secretions of important things, that is why small intestine is lined by this. You require a lot of surface area, you require a lot of absorption going on there. And if this does not work, then there is intestinal obstruction. You have to cut that thing out. And finally, you have to refix it with something. Okay. So this is the importance of histological things. You have to know what is happening at molecular level, at tiny, tiny levels. Okay. Fine. Now, there is one thing that is also given the name as this particular epithelium, right? This is also a columnar epithelium, and you can see this particular microvilli are not straight. They are having a brush border. Now, you know what is a brush border? Brush border means that the microvilli are like zigzag bristles of your toothbrush. This is particularly seen in cases of gallbladder. Okay, so gallbladder has this columnar epithelium with a brush border, right? Okay, fine. Now, finally, we finish simple epithelium. Now, if you have understood simple epithelium, understanding stratified epithelium is not a big deal because all the basic concepts of simple stacked up together form stratified epithelium. So what exactly was simple? Simple means one layer or a unicellular or a unilayered epithelium. Stratified means multiple layered epithelium. That means if I have one layer here, I keep stacking another layer, another layer, another layer, another layer. That is called as stratified or stratification, multiple layers. Fine. Okay. Now, at the same time, if you see the stratified epithelium from the top, always remember, stratified epithelium is named after the topmost layer which lines it. Suppose if a particular epithelium is shown to you and they have got multiple layers, see the topmost layer. If this particular layer you see now, this seems like squamous. Why it is squamous? Because the nuclei seems to be flat here. Right? You see a pavement-like pattern here over on the surface, and you see that they are having these dome shape, right? They are flat, they are dome shaped, they have a flat nuclei towards the surface. So this surface becomes simple squamous epithelium. Now, what is the name of this whole stratified epithelium then? If you name the topmost layer as squamous, the whole epithelium will become stratified squamous epithelium. Similarly, if you see the topmost layer cuboidal and you name the whole epithelium, it will become stratified cuboidal epithelium. Similarly, if the topmost layer is columnar, and if you see the whole layer, it will become stratified columnar epithelium. As simple as that, right? So this is how it is named. The topmost layer of this particular stratified epithelium is the one which names this kind of epithelium. Okay, right. Now, first let's see about stratified squamous epithelium. Okay, so stratified means multiple layers of what? Of squamous layers or squamous epithelium. So I told you the topmost layer has to be squamous, so that's why it is stratified squamous. Now, stratified squamous epithelium is again divided into two categories: keratinized epithelium and non-keratinized epithelium. What is keratin? Keratin is something which is hard, rough. Okay, they are secreted by the cell as a hard and rough substance. Now, where do you require such hard and rough substance? You require this kind of a substance where you require the maximum protection. So now example, which part of our body is lined by stratified squamous keratinized epithelium, which is under the most wear and tear? Correct? So imagine which is the most thing that is under wear and tear? Our skin. So outer surface of all our body skin is lined by stratified squamous keratinized epithelium. And if you see on the other side, non-keratinized epithelium is seen on those areas where the surface has to be moist, yet it has to be protective. So you need a moist surface. That means it will be under wear and tear, but still it won't be hard and rough like keratin, because here the cells are dead. When they enter, you won't see any nucleus here. See, there is no nucleus left. You can see only one layer here, but there's no nucleus left. That means these cells on top are dead. That's how we call it keratinized. But here you can see the cells are still alive till the surface. That means this is a stratified squamous epithelium, which is non-keratinized with living cells on the top, and it is keeping our surface moist. It is not keeping it dead, right? Okay. So this is the particular example of those things which are under wear and tear every day, but still they happen to be moist. How? Esophagus. You eat food every day, right? Once the food is going inside, it is scratching all the epithelium there, but it cannot be keratinized. It cannot be dead. It is non-keratinized because it is keeping it moist. Similarly, your buccal mucosa. If you see your buccal mucosa inside, it is thick. Every day you're eating food, churning food inside, but still it is helping you churn it, right? That is also lined by non-keratinized stratified squamous epithelium. Okay. Similarly, if you see the underside of your lips, that is also stratified squamous epithelium. If you see the underside of your tongue, that is also stratified non-keratinized squamous epithelium, right? So all of these things which want the surface to be moist, but at the same time they want to decrease, or basically they want to decrease the wear and tear on an everyday life, they will be lined by non-keratinized stratified squamous epithelium. Got it? Fine. Now we move on to the next category, that is stratified. Where do you find stratified cuboidal or columnar epithelium? So this will finish off stratified after this. Stratified cuboidal or columnar epithelium is seen in exocrine glands. What do you mean by exocrine? Exocrine glands means that they make something and they secrete it out, right? Just as your sweat glands, they secrete sweat. Just as your pancreas, they secrete certain enzymes, right? So they are exocrine variety of glands. They're making something and expelling them out. They are all lined by cuboidal as well as columnar, simple stratified columnar epithelium. So you see, there are multiple cube-like cells, and or they are multiple columnar-like cells later on, and they will be mostly seen in exocrine gland variety, right? So this is about stratified epithelium. Now comes the third variety, that is pseudo-stratified epithelium. Now, if you just put the same logic of stratified, what do you mean by stratified epithelium? Multiple layers. So see, multiple layers of epithelium is called as stratified. But why pseudo is here? Pseudo means false. That means I am seeing under the microscope and it seems to be that the cells are multiple layered. But actually the cells are only single layered. They are only single cells. But they seem to be multiple just as a stratified epithelium. How? Because their nuclei are at different levels. So the person who's observing this particular epithelium will feel that no, there is so many cells inside here. Is that pink and blue background? But no, this is a single layer of cell which has nuclei at different levels, making it pseudo-stratified epithelium. Okay, this is the meaning of pseudo-stratified epithelium. Now, where do you find this pseudo-stratified epithelium? This is what you need to remember. So you have got three types of epithelia which are pseudo-stratified. One, which do not have cilia. They are seen in vas deferens. Vas deferens, I already told you that this is something which takes the spermatozoa and the male urethra. Ciliated type are seen in the lungs, I told you, in the tracheobronchial tree. And the last is with stereocilia, which is nothing but a thickened microvilli. And this is seen in something called as epididymis. What is epididymis? It is a storer of spermatozoa. Once the spermatozoa are formed from the testicles, they store for some time in this convoluted tubule that is called as epididymis. So that is lined by stereocilia. This is a very important MCQ. That which of the following structures are lined by stereocilia? Stereocilia is lined in epididymis. Okay. So just to remember this mnemonic, remember two E's where stereocilia is found. One E is epididymis, another E is ear. Which part of the ear? Internal ear. Okay, so two E's are the ones which are lined by stereocerium. This is how you can remember. And once that MCQ comes, this is something that you have to memorize by the end of the day. But now you exactly know what you mean by pseudostratified epithelium. Okay, now coming to the last part of our whole histology basics of epithelium, that is something called as a special epithelium, that is called as transitional epithelium. Very, very important for your exam point of view, because this is something that usually is given to you to confuse you in your MCQs. Many times asked. Okay, so what is transitional epithelium? Transitional epithelium is an epithelium which is specialized to be there inside the bladder. Okay, your urinary bladder is lined by this epithelium. That is why we call it urothelium. Okay, because it is inside the urinary bladder. Now, what is the characteristic of this particular type of epithelium? Now, see this. One, this is multiple layered. That is the first thing that I can see. Second, if you see here carefully, the shape of the cells is very weird. It is not like stratified, which has similar kind of cells. It is not simple. These cells are of different shapes. So you can see the lowermost cells seem to be a little columnar. Above that, these cells become a little polygonal. And on top, these cells become umbrella-like. This is a characteristic of urothelium. It is lined by umbrella-shaped cells on its free surface. That means if there is a bladder, these umbrella-shaped cells are actually towards the lining of the bladder or towards the inside of the bladder. Now, what do these umbrella do? Just see this. This is the same umbrella that are here. What do these umbrella do? Umbrellas are like covers. Okay. So what does an umbrella do? If something is raining from top, an umbrella will cover us, right? Similarly, inside the bladder, there is a lot of urine. So this particular thing helps prevent the underlying cells. This is just like an umbrella cover over it so that nothing goes inside. It is a protective barrier. This is the first thing. And the second thing is, if you see these umbrellas, they're tightly stacked to each other. So what does it mean? If they're stacked with each other, once the bladder is full of urine, it will stretch out. And once it will stretch out, these epithelia, you can see they will become flat or they will become flattened. So this is the stretching power of this kind of a transitional epithelium or the uroepithelium. So another 2 points, it prevents the percolation of toxic products from the urine inside because of these umbrella shaped cells. And the second is it helps the stretching of the epithelium when the bladder is full. So it will help stretch the whole bladder. And you can see this is an unstretched bladder. This is a stretched form of a bladder where it becomes flatter, right? See, it has become flat, right? So this is the importance of urothelium or transitional epithelium. Don't forget umbrella cells. Now just see here. These are the umbrella cells. And if you carefully see inside them, they have these dot. What is this? This is the glycoproteins. These are the specialized proteins which are present in them. And what do these do? They prevent anything to percolate inside, right? So you can see in the transitional epithelium, the lowermost layer is again columnar. And you know what is columnar? They are elongated. They have got elongated nuclei at the base. In the center, these become like hexagonal. They become polyhedral. And on the top, they are these specialized umbrella-shaped cells, right? So this will again have a protective barrier against urinary products, and at the same time, it will help stretch the epithelium so that the bladder, when the bladder is full, it will help it stretch out. Fine. So this is the whole hodgepodge that we really think about in histology, and there are so many things. But what we try to do, and specifically what we are trying to do in our everyday webinars, is trying to make things simple for you so that you can think a logic behind them, right? So things are not ending here. I am going to get Ryan sir right away so that the students don't sleep away. And let's see how this particular epithelium lecture has helped you out in particular clinical scenarios. Over to you, Ryan sir.
So first of all, what a nice job done by Dr. Sanchit. Even I had forgotten a few things and it refreshed my memory as well. So really great job done. And now I think we'll ask the students a few questions and see how much they have grasped and how interested they are in this whole concept of epithelium. So we'll go to the first question.
Right.
And see how why did we study about this epithelium? Why is epithelium important? So the first question we'll discuss today is a 37-year-old male patient is complaining of pain abdomen and nausea since 1 week. No other complaints. On examination, his abdomen is soft, non-tender, with no palpable mass. He is suspected to have gastritis and is subjected to a gastroscopy. So gastroscopy is nothing but visualization of the interior of the stomach by passing a tube through the mouth. So during this gastroscopy, the camera went and saw the inside part of the stomach, and then a biopsy was taken. One small piece was taken and examined under the microscope to look for any abnormality. Now, when this epithelium was looked under the microscope, it had this appearance. So the question is, what kind of epithelium was seen? So is it simple squamous epithelium, simple columnar epithelium, simple cuboidal epithelium, or stratified squamous epithelium?
Yeah. So anyone for this particular question? Don't see the highlight because that is something that is accidentally there. So just try to understand that what exactly is wrong with the patient after that gastroscopy. Any one particular person who wants to take this?
Yes, I think there is somebody raising the hand. Afshan.
Yeah. Yes, Afshan, can you just unmute and answer this? Afshan is there? Hello? Afshan, can you hear us?
Yeah, he's not able to unmute himself.
Okay, Afshan, what do you think the answer would be? Can you write it? Second option, okay, it is simple columnar epithelium because it is present in abdomen. Okay, can you just tell me why this is, or which part of the abdomen I'm actually talking about? Where will you find this simple columnar epithelium in the abdomen?
So Afshan says stomach, sir.
Correct. So simple columnar epithelium is present in the lining of the stomach. That's right. And it has got no surface modification as such. Right? It is rich in goblet cells and it has got no more surface modification. That is why simple columnar epithelium happens to be the right answer. Absolutely right. Fine. We'll just take the next one. Okay. Now, here I just want to get in one of the students. Can you tell me by this particular thing, okay, which part of your body will be lined by this particular type of pavement-like epithelium?
Please feel free to comment the answers.
Which part of your body you will find this particular kind of an epithelium? Can anyone tell? This is pavement-like and it is single layer. So let me call it, this is simple squamous epithelium. Which part will you think where simple squamous epithelium can be present? If you remember my mnemonic. Simple squamous epithelium. Yes, somebody said that. Somebody said Bowman's capsule. Okay, I'm very happy that people have actually remembered that. So remember the mnemonic blame, right? So Bowman's capsule, alveoli, again, you've got so many things, the lining epithelium, okay? Then you've got mesothelium, and then after that, you have the endothelium and the endocardium. Correct, correct. So Ayushman, great job. And Vishal also has said alveoli, so great. That is the right answer. Okay. Now I'll just move on to one more thing. Just let me take down one more scenario with you. Okay. So now this is not going to be, you know that this basement membrane is present here. Can anyone in the comment box tell me what are the two parts of the basement membrane? Can anyone tell that? I told in the beginning that basement membrane is present. What are the two parts? Yes, absolutely, Ayushman. Correct. So Samreen has completed it. Basal lamina and reticular lamina. Great, great guys. You have got the right answer. Even Fatima has told the right answer. It is basal lamina and reticular lamina. Correct. So we have the right answers. Let's go on to the next one. Okay, so the basement membrane acts as, okay, so can anyone just tell me what is beneath the basement membrane? What is that thing that is present beneath the basement membrane? I told that. Correct. So Ayushman is fast and fastest finger first. Connective tissue is the right answer, right? So I'm just showing you, and what exactly is the question behind it? I'm asking you that. Okay. Now let me take you to another scenario. Right. Okay. Now this is interesting. I will try to dig in your memories this way. 65-year-old male patient was diagnosed to have small bowel obstruction, for which his distal or the last part of the ileum had to be resected and anastomosed to the large bowel. Now, post-surgery, his absorption of food was affected. We know that. Okay. Now, can anyone tell me what will be the lining of the small bowel or the small intestines, which has been affected? That's why the absorption of the food is gone. And that's why you had to have that kind of, okay, there is something as microvilli. Villi, okay. Can you be a little precise? Villi, microvilli is right. Can you tell me the border? Is it striated or is it brush? Absolutely right. Striated border columnar is the right answer. I'm so happy that students actually have taken and understood, and finally you have your answers, right? So correct. So it is, the small intestine is lined by brush border columnar epithelium, and that is the reason absorption of food is not happening in this particular patient. And because of that small bowel obstruction, you have to resect that out and you have to join the large intestine back to the remaining segment. Okay, fine, great. Now we move on to another scenario. Okay, so this is a continuation of the previous question. Okay, so this I think you have already told me. So this is what we have already discussed. Right, now let's move on to another scenario. Right, so this is one thing that your professor might talk about or might confuse you, and this is one of the MCQ based questions also. So I'm just going to give you a little hint here. Wherever you see this word Schneiderian, always remember nasal passage. This is a particular membrane which is, again, now can you see this epithelium here? This is what? Pseudostratified ciliated columnar epithelium. Okay, now in this only I'll get a question. Goblet cells secrete what? Goblet cells secrete what? Mucus. Correct. Mucin. Correct. So Pratik also and Ayushman also have got the right answer. They secrete mucus or they secrete mucin. And that is what is making this. So in sinuses, when you have these sinus require a lot of mucus. Correct. They are filled with mucus. So that is why sinuses are lined by this particular epithelium. Pseudostratified ciliated columnar epithelium in the sinuses and the nasal cavity is given the name as Schneiderian epithelium or Schneiderian membrane. Okay, remember that. Okay, this is an important MCQ for you all. Okay, now let's move on here. Okay, this again I'll try to confuse you with the regular. Okay, so you are brushing your teeth, and because of your vigor action, you have sparkling white teeth. You brush so hard that the bristles scrape the inner aspect of your cheek, and the inner lining of your cheek is scraped. What type of epithelium did you just scrape off? So here it is written, stratified squamous. Can anyone tell a better form of this? The inner lining of your oral cavity, the cheek. Correct. Absolutely right. This is stratified squamous non-keratinized epithelium. Correct, right? So this is how you need to make your things. Okay. Again, to confuse you, which is the hardest part of which part of the oral cavity do you think is lined by stratified squamous keratinized epithelium? Oral cavity I'm talking about. Can you tell me which of the part of the oral cavity is lined by keratinized stratified squamous epithelium? The most wear and tear and the hardest part of the oral cavity. Anyone? No, not the lips. No, teeth. No, teeth is a separate thing. No, more than that, hard palate, the alveolus, correct, the gums, right? These, if you see, they are very hard, right? You can't have that hardness over on the sides. You don't have that hardness over the floor of mouth or underneath the tongue. You have these kind of things which are hard. And why they are hard is because they are having a lot of masticatory forces again and again, friction. That is why when you open your mouth and feel your hard palate, you'll feel a lot of friction and rugae there, right? You must have seen a lot of rugae or rugosities there. It's because of this epithelium. That's why we call it masticatory mucosa. Okay, it will come later on. Don't worry. This is all going to be dealt separately in separate topics. I am just trying to ignite that thinking process inside you so that now you start examining things well. Okay, fine. Okay, so now this is what a goblet cell is. Okay, that looks like a goblet of fire. I told you it looks like a cup. Right. Now, can anyone here tell me that this mucin is secreted by which part of the goblet cell? There's another MCQ. What inside the goblet cell secretes this mucin? Granules. Okay, to be a little precise, vesicles. Correct. So Samreen has got the right answer. These are vesicles. Okay. So Pratik, these are not granules. These are vesicles, and these are secretory vesicles which are present towards the apical side of the cell, and they are filled with mucin. That's why they are called as goblet cells. Okay, fine. Right. So now comes my last question. So now see this, ignite your thinking. 63-year-old male who had an ulcer of the foot underwent split skin grafting from the thigh to cover the wound of the foot. What type of epithelium was present in the graft that was removed? So what type of epithelium do you think, if we have taken a skin and we have of the thigh and we have taken as a graft and put it there, right? So anyone? Keratinized, stratified, squamous, absolutely right. So now you know why, because it is skin. It has to have a lot of wear and tear. And now I finally see so many answers, and I'm very, very happy that you guys have finally understood the basics of histology. Now, always remember, there are always questions which are here and there. Stick to your basics first. Always remember what you have learned today. After that, you will have a lot of exceptions coming up later on. But this is what the basics is, and this will stick with you for a long, long time. Okay, don't forget that. I'm very happy to whoever has participated today and whoever has got the right answers. I think, Aishwarya, there are a lot of people who have answered closely, so it's very difficult to pick out who has answered. But I'm happy that people have understood histology. So this is what the line says: the more you sweat in practice, the less you bleed in battle. Remember that. The more you practice, the better you become. Right? Fine. So Aishwarya?
Yes, sir.
I have completed this particular thing, and I think it's over to you.
Yeah, thank you so much, sir. It was such a brilliant and amazing session. I know, like, every single detail. The best thing about you and the most important thing about you is that you give a reason to every single point that you put out, and that's what stands out when you take a class. And we love that about you when you do a class. Happy to hear your class again. Also, over to my dear students, if they have any doubts, I would just like to ask them if they have any doubts, sir, before we wind up the session.
So there's a lot of histology coming up. We are taking up every topic. So today, surface epithelium, we are going to take glandular epithelium separately. We're going to have neuroepithelium separately, muscular epithelium separately. So we have got lined things, but this is just to ignite what is histology and what exactly the basics of histology. So if anyone has understood today, then the coming lectures also you will understand.
Exactly. We have actually scheduled up the whole thing. So in case you have any doubt in this session, whatever sir has taught today, in case you have any doubt, please feel free to ask. If there is no doubts, then we can wind up the session.
So any doubts, students, you're free to ask at any particular point of time. Even if you have any kind of doubt, I would say read this topic up within this week, so that you don't, it doesn't wash away from your brain. Otherwise, it will be stuck. It will be cemented inside you once you have read it after this class. And I would suggest that if you follow Inderbir Singh for histology, there is another book which has been brilliantly written, and it is a good source for histological reading.
Sir, I think they are really well versed with today's topic, because it was such an amazing class, I must say. And time has overshot, so let's wind up very quick. Thank you so much for to everyone who has been listening to the whole session. And we'll be coming up with such amazing sessions on the other topics of general anatomy very soon. And we'll be back on Monday, 6:30 p.m. So thank you so much. See you all. Bye-bye.
Thank you very much. Thank you. Good night.